Hey everyone, welcome to this explainer. Today we're diving into something absolutely wild, bioinformatics. If you've ever found yourself wondering how on earth modern science handles the just staggering complexity of life at the microscopic level, well, you're in the right place. Think of bioinformatics as the ultimate translator for biological data. It's a discipline that takes the raw code of living things and turns it into readable, actionable insights.
But before we really get into the weeds, let's just stop and ask a pretty massive question. How do scientists actually make sense of billions of points of DNA data? I mean, modern biology is pumping out this overwhelming avalanche of genetic letters, A, C, T, G. And let's be real, relying on manual observation, that's just not humanly possible anymore. We need a massive, hyper-sophisticated solution just to read the instruction manual of life.
And that is exactly where the hero of our story steps in. So the official definition of bioinformatics goes like this: the science of developing and applying computational methods for understanding biological information. It's literally the ultimate bridge. It perfectly mashes together biology, computer science, math, and statistics so we can collect, store, and actually interpret all that complex molecular data without losing our minds.
All right, to really wrap our heads around this, here's the game plan. First up, data meets biology. Then, we'll look at the 4 pillars of omics. Third, we'll crack open the Bioinformatics Toolkit. Fourth, real-world applications. And finally, we'll wrap up with AI and the future.
Okay, Section 1, Data Meets Biology. Let's look at these computational bio-origins. Now, the historical timeline here is super fascinating. It kind of started taking root back in the 60s with early protein sequencing. But check out these milestones. We start off with Watson and Crick discovering the DNA structure in 1953. Fast forward to 1977, and we get the very first DNA sequencing methods. Then things just completely explode between 1990 and 2003 with the Human Genome Project, a total game changer. And that brings us to the present, where we are accelerating straight into an era totally driven by AI and machine learning applications.
So, to handle this absolute historical surge in data, bioinformatics has some very clear objectives. First off, it aims to store biological data efficiently. But it's way more than just a massive hard drive. It's built to actually analyze DNA, RNA, and protein sequences. It helps identify specific genes, spot mutations, predict complex protein structures, and crucially, support drug discovery. At the end of the day, all of these goals serve one huge purpose: to profoundly improve how we understand biological systems.
Moving right along, Section 2, the 4 pillars of omics. The subfields of biology.
So bioinformatics basically operates across 4 main macro to micro layers. First, you've got genomics, which is literally the study of entire genomes. Then proteomics, focusing on proteins and what they do. Going a bit deeper, we hit transcriptomics, studying the RNA transcripts produced by our genes. And finally, metabolomics, which dives into the metabolites inside biological systems. Let's look at what scientists actually do with these pillars.
Starting with the first 2, genomics and proteomics. Over in genomics, scientists are using computational tools for things like whole genome sequencing, pinpointing specific genes, and doing comparative genomics between entirely different species. Meanwhile, proteomics lets scientists predict actual protein structures, figure out how different proteins interact with each other, and discover vital biomarkers.
Then we've got the other 2 pillars, transcriptomics and metabolomics. With transcriptomics, researchers run gene expression analysis and functional genomics, both of which are absolutely crucial if you want an accurate disease diagnosis. Metabolomics steps in to identify disease biomarkers by mapping out complex metabolic pathways. These molecular changes are very often the very first red flags of how a disease is operating inside the body.
All right, Section 3, the Bioinformatics Toolkit. Processing the data.
To understand the toolkit, you really have to look at 2 sides of the coin: storage versus action. On the storage side, we have biological databases. Think of these as massive digital libraries, like GenBank, that just hold biological info like DNA sequences. But on the action side, we have bioinformatics tools. This is where things happen. Software like BLAST doesn't just sit there holding the data, it actively analyzes and compares those sequences so we can actually find meaningful patterns.
Let's quickly highlight a few of those foundational storage banks where all this info lives before the real analysis starts. I mentioned GenBank, which is this massive public database specifically for DNA sequences. Then you have the Protein Data Bank, or PDB, which holds 3D protein structures. And of course, scientists rely heavily on UniProt, which is basically the go-to, comprehensive database for protein sequences.
Okay, once the data is safely stored, it's time to bring in the heavy machinery. Let's do a rapid-fire workshop of the software tools. First up, BLAST. Honestly, it's essentially the Google search engine for biology, used constantly for comparing DNA and protein sequences. Then we have Clustal Omega, the ultimate tool for aligning multiple sequences all at once. Mega steps in for evolutionary or phylogenetic analysis. And finally, BioEdit, which is the vital software for sequence editing.
Up next, Section 4, Real-World Applications, the Data Impact.
When you actually look at the applications, the sheer diversity is just staggering. Through genome analysis, we can quickly detect mutations. In drug discovery, bioinformatics makes virtual screening and molecular docking possible, meaning scientists can test a drug against a target entirely on a computer. It's also the backbone of personalized medicine, tailoring treatments using pharmacogenomics. Even agriculture uses it to create disease-resistant plants, not to mention its huge role in industrial biotechnology and synthetic biology.
But let's make this deeply personal for a second. In disease research, bioinformatics is quite literally a lifesaver. It is the primary way modern medicine identifies disease-causing genes and those crucial cancer biomarkers. It powers infectious disease surveillance, uncovers how drug resistance actually works, and is absolutely essential for diagnosing super-rare genetic diseases. It basically takes an invisible enemy and turns it into readable, trackable code.
And the practical advantages here are just immense. Bioinformatics effortlessly handles massive biological data sets that would otherwise be totally unmanageable. Because we can run virtual screenings and rapid sequence comparisons, it drastically cuts down research time and costs. It boosts the accuracy of analyses, builds the foundation for personalized medicine, and beautifully facilitates global data sharing among scientists everywhere.
Now, we do need to balance the picture because this field definitely faces some serious hurdles. First off, simply managing datasets of this sheer size requires incredibly advanced computational power. There are also very strict data security and privacy concerns, especially when you're dealing with human genetic info. Plus, biological systems are just wildly complex. And finally, you can't just be a biologist or a coder. You really need strict interdisciplinary expertise across biology, math, and computer science.
Finally, let's head into Section 5, AI and the Future. What's next on the horizon?
Unsurprisingly, artificial intelligence and machine learning are stepping in as the ultimate accelerators here, pushing data interpretation into absolute overdrive. We're seeing AI take center stage for incredibly complex tasks like predicting protein structures, identifying patterns for early disease prediction, radically speeding up drug discovery, and handling deep genomic data analysis. AI is completely reshaping how fast we can interpret life at the molecular level.
Looking forward, the prospects are nothing short of transformative. We are speeding towards a reality dominated by AI-driven genomic analysis and precision medicine. We're going to see bioinformatics providing advanced support for gene editing technologies. Ultimately, these innovations in large-scale biological data integration are going to completely transform personalized healthcare, along with agriculture and biotech.
To summarize our explainer today, I want to leave you with this really powerful takeaway straight from the source material. As biological data continues to expand, bioinformatics will remain a cornerstone of modern scientific research and innovation. It really is that vital bridge between the biological and the computational, turning raw sequences into actual real-world breakthroughs.
Which leaves us with a pretty provocative thought to end on. Knowing that these computational tools are unlocking our genetic code right now, and with AI driving precision medicine faster than ever before, how personalized do you think your own healthcare is going to become in the next decade? The code is already being written. Thank you so much for joining me on this explainer, and keep exploring.